This meeting is being recorded.
Just in case. Oh, now everybody's faces went away. As soon as you hit record, boom, all gone. But thanks so much for coming out tonight, everybody. I'm very excited about this series. The Pick a Brain, Bird Brain series this year is all highlighting Hawk Ridge research efforts. There's some exciting stuff happening, and I'm really delighted to have met Etterson, who's our chair of our research committee for Hawk Ridge, who's been doing some amazing work. And we're going to hear about PFAS tonight, but I wanted to, since Dick is still showing his face, give a brief shout out. He's going to come on next month on the 21st of March, and we'll be hearing about northern goshawks from Dick. So I'm excited about that. And each month we're just highlighting some of the amazing work that's been happening. So yeah, I think without doing a whole lot of carrying on, I'm just going to go ahead and make Matt the host so he can, yep, I do want to make Matt the host. So he can take over the screen and we can go ahead and get started. PFAS is one of those things that is hitting the news in a big way right now. And I'm really excited, like I say, to hear a longer presentation about PFAS and birds with the cooperative program between well, I'm going to quit stealing thunder and let Matt tell us about all of that kind of good stuff. Thanks so much, Matt.
Okay, thanks, Margie. Really grateful to have the opportunity to talk to you all today. Give me a moment to try to figure out how to share my slides here.
Looking good.
All right. I thought I'd start off by acknowledging this Eagle Cam photo, which was sent to me this morning. This was taken this morning down in the Twin Cities area of a bald eagle sitting on eggs right now as we speak, or as we spoke back then this morning. So hopefully those of you who are in the path of the great blizzard of 23 are sitting as comfortably as she is right now and can sit back and listen to this talk with me. I am going to make the assumption that many of you are new to PFAS. As you'll see as I scroll through my slides here, I've tried to use as many open source and public sources of information and provide URLs for those of you who want to learn more. And so hopefully this will be a resource that will be something you can come back to and follow up on for learning more. Let's start by attempting to define these substances. So PFAS stands for polyfluoralkyl substances, and that's why we call them PFAS. If you look at the tree on the right-hand side, you'll see that even the definition of these things is quite complicated and can be a little bit overwhelming. So I'm not going to dwell on the chemistry or the nomenclature really here. I'll simply tell you that these are, as it currently stands, considered to comprise over 9,000 compounds that do not occur naturally. And I'll very colloquially be referring to these as chemicals with essentially a carbon structure to which fluorines are attached, and often with some kind of a functional group on the end. And to give you an example of that, here are a couple of diagrams of one particular PFAS chemical. This is PFAS. Some people call it PFOS to help reduce the confusion with PFAS, but this is perfluorooctane sulfonate. It's perhaps the best known of the PFAS, and I can walk you through the nomenclature a little bit here. So the oct part, the OCT, refers to 8 carbons in the carbon structure. The perfluoro part references all of the fluorines attached to those carbons, and the sulfonate part of the name refers to the functional group or the head of the molecule. And while these molecules can be quite diverse, the carbon-fluorine relationship is really what I will be referring to. And it's also what lends these chemicals their persistence in the environment. The carbon-fluorine bond is very strong and makes these chemicals very persistent. Now, this particular chemical has been mostly phased out of production, but it is still nevertheless the one we know most about, and also remains for the most part the PFAS that is most commonly observed in avian tissues.
Matt, I've gotten a comment in the chat from someone that says that your screen is not shared. I can see it. Allie says she can. So, yeah, okay. So don't worry about it. Looks
Okay,
like
well, I'll carry
most-
on. Whoever you are, I'm very sorry. I hope that if you can't get it sorted, you'll be able to watch the recording after the fact. So on this slide, I kind of wanted to walk through the developing awareness of PFAS in drinking water in the United States. And so our early awareness really occurred in 1999 in a town in Parkersburg, West Virginia. If any of you have seen the movie The Devil We Know, this is the town and its environs were the subject of that movie. And since then, if you look across these maps of the United States, you'll see our awareness and understanding of PFAS in drinking water supplies or potentially contaminating drinking water supplies has increased considerably, particularly in the Northeast United States over those 2 decades. Now, as that awareness has increased, our understanding of the toxicity of these chemicals has also increased. And if you look at 2 common PFAS, PFOS and PFOA, and the evolution of the Minnesota guidance for safe concentrations in water, you'll see that the limit for safe concentrations has dropped by over 2 orders of magnitude in the guidance. And so we've really become more aware of the potential toxicity of these chemicals over the same time frame. So you might now be wondering, where is this stuff coming from? Well, as I mentioned before, these are all entirely manufactured chemicals. They come from various industrial processes, from which may, the industrial processes themselves may produce waste releases to the air or water, or they may result in the creation of products that then perhaps end up in landfills, which also contributes to both air and water contamination. And one major use of these chemicals is in what we often refer to as AFFF, or aqueous film forming foams, that are very useful for fire suppression. And so very often for fire suppression, these are sprayed into the environment, and so then get into the water and soils that way. And so while I won't be focusing on human exposure here, I will be focusing on environmental exposure, and particularly subsequent exposures to birds. Landfills, as I hinted over here, also have been shown to be a source of PFAS in the environment. And these are closed landfills in the state of Minnesota, with leachates exceeding Minnesota health guidelines for PFAS. Now, I will add a cautionary note here. The fact that the leachates exceed Minnesota health guidelines does not necessarily mean those leachates are getting into ground and surface waters. Many of these landfills will be lined, and some of those leachates will be brought to processing plants. So don't get too alarmed about this, but this does indicate that landfills can be a potential source of PFAS in the environment. Once in the environment, PFAS partitions to a variety of matrices. It gets into the soil, gets into surface and groundwaters. Once it's in surface water, it can partition to the sediment, and it also can be volatile, so it can get into the air. These chemicals can also be transported quite long distances in both air and water. And in fact, studies have shown that both Arctic birds and mammals have been exposed to PFAS chemicals. So this leads to a question from those of us who are ornithocentric. Are PFAS a problem for birds? And if so, what species? Toxicity testing suggests that birds may be a sensitive receptor to PFAS, but birds, of course, are a very diverse taxon. They occupy a wide variety of habitats on all continents and eat a great variety of diets. So perhaps it makes sense that not all birds would be equally exposed or equally sensitive to these chemicals. And so a major question is whether we can begin to figure out which birds are likely to be exposed and which ones will be most sensitive. And so here I've broken this major question down kind of into three sub-questions. And up to now, I've really been talking about this first question: what is the environmental distribution of PFAS? Where does it come from? And where does it partition and distribute once it's in the environment? The next question, which I'll kind of pivot to now, is to ask, given the environmental distribution of PFAS, what species, in particular, what bird species are likely to be exposed? And then, of course, knowledge of exposure would lead us to ask the question, given exposure, what species are most likely to be affected? Now, we're going to make the assumption here that diet is the major exposure pathway for birds. Now, there's some evidence that drinking water may be a route of exposure, but I'm not really going to be talking about that too much tonight. Diet does seem to be the major route of exposure. So, what species are most likely to be exposed? Well, some PFAS are known to bioaccumulate and biomagnify. And I'm not really going to dwell on these two processes, except to say that working together, they result in animal tissues having a much higher concentration than the environment in which they live. For example, the graph on the right-hand side shows what are called bioaccumulation factors, which is simply the ratio of PFAS in animal tissue to the water, in this case for fish. And you can see that the median bioaccumulation factor for freshwater fish is that they have about 1,000 times the PFAS in them than is in the water in which they live. And so this potential for increasing concentrations into animal tissues can result in a process where animals feeding at very high trophic levels may experience much higher exposures than those feeding at lower trophic levels. And in particular, piscivorous birds may be at risk because of this known increase in exposure in fish. So initial concerns over PFAS in birds indeed focused on piscivorous birds. And here I'm showing you measured concentrations of PFAS. And I'll pause for a second here and talk about the concentrations. In all of the graphs that I present tonight, they will all be in parts per billion to try to make them as comparable as possible. But they're not all exactly the same. And that's because the per billion part changes. In this case, these are concentrations in tissues. So they would be parts per per gram of nanograms per gram of tissue, for example. But we might also see the denominator being water or some other measure. And so they'll all be parts per billion, but be a little bit careful in interpreting that. That doesn't necessarily mean that all these numbers are comparable to one another. So anyway, you can see here among these groups of birds classified by guild that there's a wide range in measured concentrations in their tissues that ranges over at least 3 orders of magnitude. And indeed, some fish-eating birds, both seabirds and waterfowl or terrestrial fish-eating birds, do have very high exposures, but some do not. And in fact, there's some tantalizing hints here that some insectivores or non-piscivorous water birds may also be experiencing exposures. So these kinds of observations led us to wonder at Hawk Ridge what exposures have terrestrial birds of prey experienced, and do those exposures differ among species, and if so, why? Well, Hawk Ridge seemed like an ideal place to ask these questions. As many of you know, probably all of you know, we are the focus of one of the great migrations in North America as birds of prey come down out of the northwest part of the continent, circle over top of the Great Plains, and follow the edge of the boreal transition forest until they encounter Lake Superior, at which point they turn and funnel past Duluth, where we have observers positioned at our count station on the left here. You can see them looking out over Lake Superior, where we count over 70,000 raptors per year. And we trap between 3,500 and 4,000 raptors every year. So we thought that this trapping data stream could be a great source of material for looking at a very sort of wide global pattern of distribution of exposures to these chemicals. So to do this study, we partnered with the Raptor Center down in the Twin Cities. And one of our major questions was whether or not raptors that we were sampling coming out of the northern and northwestern United States had different tissue concentrations than birds that were being admitted as patients at the Raptor Center. Now that question got very challenging to answer because as soon as we started this study, COVID hit and shut down the Raptor Center for a period of time and made it very difficult for us to collect the samples that we needed. Nevertheless, we do have some data that I'll circle back to try to help address that question. And so in the samples we collected, up to 33 PFAS chemicals were quantified in 355 individuals from 11 species. Now, I'll quickly give a shout out to Frank Nicoletti, with whom I worked very closely in this study, who directed all the trapping and banding that occurred as part of this project. And also Dr. Julie Ponder from the Raptor Center, who oversaw the sampling down in the Raptor Center. So let's take a quick look at the species that we sampled. And I've grouped them into broad taxonomic classifications here and tried to give you some information on diet, taxonomy, and habitat that these species use. And please keep in mind that these are pretty rough. When I say that a raptor eats birds, that doesn't necessarily mean that it won't eat mammals and vice versa, but rather these are sort of broad tendencies. And the same can be said about the habitat classifications. But I'll start here with our accipiters and northern harrier, which offer a nice gradient between bird consumers and mammal consumers. And this was of particular interest to us because a previous mercury study that we had conducted had shown that raptors that eat birds had elevated concentrations of mercury in their blood compared to raptors that consumed mammals. And we wondered whether a similar pattern might be observed for PFAS chemicals. Our next group that I'll mention is the Budioninae, or the soaring hawks, lumped in with bald eagle. On the left-hand side, these are more mammal consumers. And on the right-hand side, of course, bald eagle is consuming fish and carrion, and tends to live at the aquatic terrestrial ecotone. And so those two characteristics made us think that bald eagle was a very good subject of study here. Plus, we were aware of Bill Rowdy's previous work. Bill is on the call tonight, so please, Bill, do correct me if I misrepresent that study in any way. But also we were interested in the budios on the left-hand side, in particular rough-legged hawk. The big eruption of rough-legged hawks migrating through Hawk Ridge happened during the course of this study. And this seemed like a great chance for us to compare a very northern species and see what kinds of residues it might have compared to some of these other species. And then our final group, or two groups, will be the owls and falcons. So we sampled 2 owls, northern saw-whet owl and great horned owl, and 2 falcons, Merlin and peregrine falcon. And you can quickly scan through the diets and rough habitat classifications for these species. So because this was an opportunistic study, our sample sizes are not necessarily balanced in all cases. We took what we could get, especially from those relatively rare species like peregrine falcon or Cooper's hawk. We worked hard at beefing up our sample of those. Bald eagle was by far our largest sample, followed by red-tailed hawk. Of the species that occur at both the Raptor Center and Hawk Ridge, red-tailed hawk is really the only one where we felt we have really pretty good coverage for both locations. And so for comparing the 2 locations, I'll be talking mostly about red-tailed hawk. On the minimum sample size side, we have 10 northern harriers and 10 rough-legged hawks. And please do memorize those acronyms at the bottom. I'll be showing you a slide in a moment that doesn't have the names, so get ready. So here is the distribution of the 13 major PFAS chemicals that we found in raptor plasma in the study. And you can see that PFAS was by far the most commonly observed and the chemical at greatest concentration across all of these chemicals. And this includes all species sampled. And so this is the chemical that I started out with an example chemical, and we'll continue to talk mostly about PFAS throughout the talk today. Okay, so here are species-specific distributions of PFAS in plasma. And I'll quickly walk you through these box plots if you haven't seen them before. The black line in the middle of the boxes for each species tells you what the median value of PFAS concentration, which is indicated over on the left-hand side on the vertical axis. Now, this is a logarithmic scale, so you can see those numbers grow rapidly as you increase in height along that scale. And then the box surrounding the median line gives you the first and third quartile of the data. So you can think about that box as representing the middle 50% of the data. So the majority of the central portion of the distribution of residue concentrations for that species is in that box. And then the whiskers or the tails and the dots that you see represent the extremes of the distribution, the upper 25th percentile and lower 25th percentile, respectively. And so, you know, if you look across these box plots across all these species, maybe it's a little bit underwhelming, right? We see not a lot of huge differences among species, with a couple of exceptions there. The sort of median plasma concentrations here are at around 10 parts per billion, with a couple of species showing elevated concentrations. Bald eagle, as was expected, had higher concentrations than many of the other species, but Cooper's hawk, the green box on the left there, was not necessarily expected, and that was a bit of a surprise to us. And I'll come back to that, to Cooper's hawk in a little bit. When we looked at adults versus juveniles across all species, we saw surprisingly virtually identical distributions. And so this really goes to that question of bioaccumulation and biomagnification, but especially bioaccumulation. We do not see necessarily increasing tissue residues as the birds age. And this may be because they can eliminate it through urofeces or through biotransformation. But it does not look like birds, for the most part, have increasing concentrations of PFAS as they age. Here's back to the red-tailed hawk using the comparison between birds sampled at the Raptor Center versus at Hawk Ridge. And here we did see higher concentrations in the Raptor Center patients than we observed at Hawk Ridge. And this may be an urban effect due to the birds presenting at the Raptor Center, probably having a more southerly, more urban distribution. It is probably not a PFAS effect leading to increased rates of hospitalization among these birds. I don't know that for sure, but that's a personal opinion there. But the urban effect is interesting, and I'll return to that in a moment. Now, I have several slides in a row that will all be quite similar, showing you comparisons of what we found for a given species to other data available in the literature, wherever I was able to find it. And these data will be expressed in different units depending on what was available in the publication which I found. So, for example, the Norwegian publication here gave the mean and standard deviation, and so I calculated that for the Hawk Ridge data for northern goshawks. And you can see these distributions are broadly similar. They largely overlap. Perhaps the red dot is slightly higher than the black dot, but the variation around that is very high, and I wouldn't draw any inferences from that. Here's some peregrine falcon data comparing Hawk Ridge birds to birds sampled in rural and urban Ontario. And again, there's really broad overlap in the distributions, perhaps a hint of a slight elevation in urban environments compared to rural, but again, more overlap than increase there. Here are some data from Cooper's hawk, and these comparisons, I'll warn you, are getting more difficult to make. So I ordered these slides intentionally here. The data from Hawk Ridge are in blood plasma. The data for Cooper's hawk from Vancouver was taken from eggs. And so there's a question about whether those tissues are comparable to begin with. But the egg data were further expressed in terms of nanogram per gram protein. So again, as I mentioned, parts per billion, but this time normalized to gram protein. And I reversed the transformation for those data. So the two red dots here are exactly the same data. They just represent me reversing that transformation, taking out the protein normalization and expressing those concentrations in nanogram per gram wet weight for the eggs. But again, it's still eggs and plasma. But you can see, we typically expect eggs to have higher concentrations than does plasma due to maternal transfer. But again, there's still broad overlap in those two distributions. And for any of you who are wondering, I think I did probably get that standard deviation wrong in the backwards transformation, but I didn't have enough information in that publication to get it right. So I wouldn't make much of that standard deviation, but I think the mean there is probably correct. And then the last comparison I'll make is to Bill Rowdy's bald eagle blood plasma PFAS. And I think these came from nestling birds sampled in the locations indicated in the map on the right-hand side. And I've taken the Rowdy and colleagues data and plotted them with the Hawk Ridge data, giving the geometric mean for the dot and the minimum and maximum concentrations for the ends of the bars there, as reported in the Rowdy et al. Paper. And I've tried to color code them in the same way, or at least as close as I could get in R, to the color codes used in the map on the right. And so the red here pertains to the Apostle Islands, the brown, the rusty brown here, the South Shore of Lake Superior. The blue is the upper St. Croix River, the yellow, the lower St. Croix, the purple, the Mississippi River in the Twin Cities area, and the green is the Mississippi River pools 3 and 4. And you can see, I think that our data show that PFAS concentrations are probably lower, even though there is some overlap in the Hawk Ridge birds, than reported from these nestlings. And that's interesting from a couple of perspectives. There was about 10 to 15 years separating these data. And, Rowdy and colleagues suggested that PFAS was already starting to decline in some of their data. And our data from 10 to 15 years later probably confirms that. And this is probably a result of the discontinuation of production of PFAS in the area. So that's all the exposures, the comparable exposures I could find in the literature. And so, we'll return to these questions I asked at the beginning. What exposures have terrestrial birds of prey experienced? Well, they have about 10 parts per billion background tissue residues, but these residues don't really reflect direct exposure, and we'll talk more about this in a minute. What they represent is some dynamic, perhaps equilibrium between exposure in terms of consumption of diet with PFAS in it or PFOS in it, and any elimination or biotransformation that bird is able to accomplish. But nevertheless, it does show us what the sort of background rates of tissue concentration is among the birds sampled. And so does exposure differ among species? Yes, but not in relation to age, diet, or taxonomy. But perhaps there's a hint of an urban signal in the data. And to further explore this, I created another box plot where I asked someone in my lab to arrange these species according to their propensity to nest or the extent of their range overlap with human habitation. And this is what they came up with. I didn't tell them why I wanted to do that. And then I took their classification and plotted the box plots for each of the species along that gradient. And you can see, if you kind of squint at this and look, there's maybe a hint of an urban signal in exposure, which is really interesting if it's true, because it would indicate that a major predictor of exposure is simply proximity to us, proximity to humans, and PFAS use and production. Okay, so that's exposure, and that's the Hawk Ridge study. But you'll notice that I've been fairly cagey about saying whether any of this matters. And the short answer is we don't really know yet. And I think this is a major next step in understanding the toxicity of these species to birds. And in the next few slides, I'll talk about a little bit about what we know and about another study we're conducting to try to understand the toxicity of these chemicals. So in order to do this, I have to go through some toxicological terms. Many of you will have heard of the LD50. If not, it is the median lethal dose, or it is the dose of a substance or chemical that is modeled to kill, that is thought to kill 50% of tested animals. We also have the low AL, or the lowest observed adverse effects level. So this is in a test where birds or other animals have been given a dose, different doses of the chemical. This is the lowest dose at which some adverse effect was observed. And then toxicity reference values and predicted no effect concentrations. I'm not going to describe those in detail, but these are numbers that are extrapolated from the toxicity test results and are meant to represent values that should be protective if concentrations are below those concentrations. So let's shift over and look at the graph on the right from Newstead et al., where they plotted different avian effects concentrations from toxicity tests. And one of the things you should notice right away at the very top there with the LD50 of juvenile mallards is that it takes an awful lot of PFAS to outright kill a bird. Most of the acute effects are up there at very high concentrations. And so the effects that we're likely to observe and be most concerned about are going to be what we call chronic effects. And in particular, where effects, the lowest effects observed seem to be effects on reproduction, as you see down at about the 1,000 parts per billion in the diet. Now, it would be really tempting to just go back to those numbers that I just gave you from tissue concentrations and say, oh, where do we fall on this graph? Are we below the predicted no effect concentrations or the toxicity reference values? Then we're safe. Are we above? Then we're not safe. But we can't do that. And there's a number of reasons we can't do it, but the most important is that these are parts per billion in the diet per day. So this is dose per day. And we don't know how dose per day in consumption relates to quantities in the tissues that were measured. And so that remains a major uncertainty in interpreting the kinds of residue data that I've just been showing you. Now, this is a slide that I like to show when I talk about birds and toxicity. So I'll walk you through it quickly. The tree diagram on the left hand is a very simplified phylogenetic tree for the birds, showing the very ancient birds, ostriches, emus, etc., on the very bottom bar, followed by the split between the gallo-ansares, comprising the landfowl and waterfowl, with the rest of the birds. Their split with the rest of the birds occurred about 65 to 70 million years ago. And these lines have been extended into these two tables here with two columns. The species column tells you the number of species in each of those groups among all birds, and the percentage gives you the percent of all birds represented by those species. In contrast, the next column tells you the percent of toxicity test results for those birds within the general EPA Ecotox database. Now, those are toxicity test results from all chemicals, not just PFAS, but it gives a very broad picture and understanding of what birds we're testing. And you can see there's a very strong asymmetry here, that the birds that we are testing, so about 95% of ecotox results come from this Gallo-Anser's clade, which comprises about 5% of bird species. And about 95% of bird species in the neoaves have only about 6% of toxicity test results representing them. And in particular, that's where our raptors fall in the non-songbirds line of that neoaves. And so my hope is that with this next study, we can contribute some information to filling out some of that missing data. Now, if you think, what's 65 million years? That's no big deal. Well, a lot of evolution can occur in that time. And in fact, this is what our ancestors looked like about 65 million years ago. So a lot can change. So a few years ago, as the PFAS tidal wave started to hit EPA, colleagues and I sat down and thought, well, what would an ideal avian PFAS study look like? And in designing this study, we drew on some existing, really great existing work that had been done by Chris Custer and others at USGS. And we leveraged that to develop a study that would look at exposure, so a lot of the things I've been talking about, but would also quantify exposure in birds, in the environment, in the food web, in the diet of those birds, and then look at their trophic status, how high, at what trophic level are they feeding in a food web. And then on the effects side, we would look at reproductive effects, because that's where we think the action is likely to be with birds exposed to PFAS. We would look at physiological pathways of effects, otherwise known as adverse outcome pathways in the ecotox jargon. So this is probing the kinds of physiological effects that PFAS may cause. And then we would try to validate all this using in situ dosing experiments where birds are actually exposed to the chemicals in the field at known doses to see if what's being observed in the field is replicated. And then hopefully we would put all this together using clever mathematics and modeling and be able to predict what's going to happen to birds. So here's a schematic of that study. We have 7 sites here in the Duluth Superior area. You can see that we have some sites designated as suspected PFAS impacted sites, other sites that are there as reference or experimental sites. The environmental releases that we're interested in here are largely through AFFF, or aqueous film forming foams used for fire suppression. Right over here on the right-hand side, I'm showing you a satellite photo. This is just off of Google Maps, showing you a training facility associated with the Lake Superior Emergency Response Training Center, which is close to our Boy Scout site here. We also have a site at the air base where AFFF have been used to train folks to put out airplane fires. And then we have connected waterways through a Martin Road drainage into Rice Lake. So these are our study sites. Boulder Lake is a reference site where we didn't think any exposure would occur. And we put up nest boxes for 3 species, tree swallow, house wren, and black-capped chickadee, that form sort of a gradient of diet from the more aquatic species being tree swallow to the more terrestrial species being black-capped chickadee. And all use the nest boxes, and they have different modes of feeding that may be important in explaining their exposure. And so we are kind of using these birds as a natural experiment, their variation in life history, to tell us something about how PFAS may be moving into avian food webs. So we have about 220 boxes at these 7 sites that are monitored from May to August. Boxes are checked twice a week to see if there's anything in there. If so, what species, the number of eggs and or nestlings. We record the egg hatch date, age of nestlings, and so on. This past summer, we had about a 40% occupancy rate of those 220 boxes. Most of those were tree swallows, followed by house wrens, and just a few black-capped chickadees. And of those occupied boxes, 83% produced some young to fledging. We take samples from both eggs and nestlings for PFAS quantification. We also conduct food web characterization at all of these sites, starting with the plants, moving up through the invertebrates into fish. And then on the right-hand side, you see structures with PFAS passive samplers on them that are placed in these waterways. We also combine, collect grab samples of water, soil, and sediment for PFAS quantification. And here I'm showing you just some preliminary data from those sites. These data were analyzed by our collaborators at USGS. I'll read these off to you in case they're too small. On the left-hand side is Boy Scout Landing, followed by Air Base, Martin Road, Rice Lake, Boulder Lake, and UMD. And the 3 panels show you 3 different biological samples, eggs and nestlings of tree swallows, and then the diet of those nestlings. And you can see a fairly congruent patterns across our suspected impacted sites. This pattern was a little bit of a surprise to us to see that Martin Road and Rice Lake had potentially higher contamination than did, or higher exposure than did the air base where we believe the releases occurred. And that's an interesting pattern that we're following up on. But it was also borne out in the diet of the birds. And though we didn't have reference samples of diet from Boulder and UMD, I'll point out that our egg and nestling values were quite low at both of those sites, suggesting that they probably will serve as pretty good reference sites. So I want to focus a little bit more on the eggs that were tested. This is the same box plot as I showed you in the previous graph for eggs. And the point I want to make here is that the concentrations that we observed at our suspected impacted sites had concentrations in the range at which effects on hatching have been observed or reported in tree swallows previously. And I'll point out that this was a correlational study. And so I will caution you that correlation is not causation. And in fact, subsequent work by the same authors at other sites has shown similar contamination levels without these effects. So there's some question about this dose response, but our contamination levels, our exposure levels in eggs and nestlings are certainly commensurate with what's been seen at other contaminated sites around the country. I'll return to that point in a moment, but first I wanted to show you the slide of their diet. So this is DNA metabarcoding of the tree swallow diets. This is certainly good news to any of you who live in northern Minnesota. These birds are eating annoying insects. They really like flies, and yes, there are mosquitoes in that group that they are in fact eating. But what's most impressive about this tree map, I think, is that they're eating a staggeringly diverse diet. This is about 20 broods of mostly tree swallows. There's one house wren in these data. And these birds are consuming 21 orders of invertebrates from 86 families, 220 genera, and 308 species. So these are very generalist predators. And this ups the level of difficulty for us in tracing how these chemicals might be moving through these food webs and into these birds. So back to our sort of study design slide here. I've been talking almost uniquely again about this left-hand side and exposure. I've shown you how we're quantifying PFAS in birds and in the food web and environment, how we're characterizing their diet. I haven't talked much about trophic status, but our samples have gone out for isotope characterization, or will go out soon. And so we should have information on the trophic status of these birds to relate to potential bioaccumulation. We are also conducting effects assessment, and this is the work that we're really kicking off now. Some of it is going on this winter in the lab using tissues that were collected last summer. We have reproductive effects data on the various nests that we were following last summer. We're using the tissues that we collected to inform adverse outcome pathway development to understand the pathways of effect that may be operative in these birds. And this summer, we will kick off our in situ dosing experiment. So we're just at the beginning of a lot of this work, but I think there's really great things to come. And I will point out that we are not by any means the only group doing this. And I'll direct you sort of in my penultimate slide here to the CERDP website. This is the Strategic Environmental Research and Development Program. And you can look across the years here and see what projects these funded. Each of these boxes is not a project, it's a group of projects. So ours falls within this highlighted red box here with a number of other avian studies. But if you have time to look through this, you can learn an awful lot about PFAS and their use, remediation, alternatives, effects on birds and other ecological receptors by going to this website. So a lot of really great work going on. And our project is one among many of these. And so I think in the next few years, we'll be poised to learn really quite a bit about the effects of these chemicals on not just birds, but on other animals as well. And that's really it. I'd like to just pause for a moment and thank everyone who has been involved in this study, both our funding organizations, supporting organizations, and all of the people who help collect the data and do the analyses here. It really is an army of folks, of very good folks. So thank you all. And with that, I will stop sharing and see if we have time for questions.
We sure do. So yeah, we don't have any submitted in the chat. Nice job, folks. I forgot to tell you to wait till the end to ask your questions. Well-trained group of Zoom watchers. So yeah, go ahead. I think we're a small enough group that we can handle if you unmute yourself and go ahead and ask questions verbally. I think that's fine. If you don't want to be verbal, you can go ahead and stick them in the chat. Okay.
How do you sample the eggs? So is that non-destructive sampling?
No, it is not non-destructive sampling. We collect the eggs.
I have a somewhat mundane question. I was intrigued by the slide about the diets, all the different insect and other invertebrate families. Well, how are you collecting that information? Are you somehow evacuating the bird's stomachs and the remains are clear enough that you can identify them? I'm intrigued.
Yes, good question. So this is a sensitive question. We collect nestlings. They are euthanized humanely. And after euthanization, we open up their stomachs and collect the stomach contents. We also take many other tissues, various tissues. We take thyroids, we take serum, we take livers, and all is used for our adverse outcome pathway characterization.
Thank you. Well, I'll volunteer another question then. I'm intrigued that bioaccumulation seems to level off in these birds. As you suggested, they may be metabolizing them or passing the waste in some fashion. So in that respect, it strikes me as being different from chlorinated hydrocarbons. And as a result of that, do you think the adverse effects might be quite different?
That's a really great question. And there's an awful lot to unpack in that question. We don't fully understand the bioaccumulation mechanism for PFAS yet. They seem to have an affinity for proteins, and in particular, proteins found in blood serum. And that is part of the bioaccumulation puzzle here. That's quite different from the chlorinated hydrocarbons, as you probably know, given your awareness. They partition to lipids and to fatty tissues. And so that's a different route of bioaccumulation, if you will. The bioaccumulation picture for these chemicals is further complicated by what's called enterohepatic recycling. So kidneys are in many cases able to remove these chemicals and shunt them into urine on its way out, but then those chemicals are subsequently reabsorbed back into the bird. And so that's where the recycling part occurs. And so that's also potentially part of the bioaccumulation problem with these chemicals.
Yeah, but it intrigues me as being very interesting. Apparently, they don't hang out in the fats for a long period of time, such as DDT or DDE would, and that way aren't available for the bird to access in periods of stress and whatnot.
Yeah, I think you're correct about that. The female birds are also very good at dumping these into their eggs. And so that's part of the reasons why we see eggs having very high concentrations of this. So they are, at least female birds have that route of elimination.
Thank you.
So Matt, correct me if I'm wrong, but are you taking samples? Is it blood samples or is it the whole body sample?
So it depends on the study. For the raptors, we were taking blood samples. So we were sampling up to 2 mils of blood and then spinning that down to get the plasma. And the plasma was what was sent for analysis. For the songbird study, we were collecting whole organism.
Yeah, so the results would kind of indicate different things. I mean, blood is going to be the latest meal or series of meals within 24 hours or something like that, whereas the organism is going to be whatever is shunted out into the rest of the organs of the organism and not passed somehow. So kind of different levels in those tissues, right?
Yeah, your logic makes sense to me, Bill, and I don't totally have a response. It wouldn't surprise me if birds can mobilize PFAS from other tissues into their blood. So I don't know the extent to which blood represents the most recent meal. It may, but I don't know the extent that that's true. But I agree. The logic makes sense here.
One of the things, since I'm asking the question now, is that your one slide showing that you've got more concentrations of PFAS at a lake that is downstream, I think, if I recall you saying it, from where you thought the release site was at an air base or something like that. I might have this quite a little bit wrong, but it seems to me that I just wonder whether or not the PFAS chemicals were flushing downriver towards that lake and then settling in that lake so that there were more bioavailable in that lake compared to the release site.
Yeah, great point, Bill. We think that's one of our hypotheses for what's going on there. The other hypothesis, which I really didn't discuss today, is that there is a closed landfill adjacent to that lake that may also be contributing to PFAS levels in that lake. Now, those two hypotheses aren't mutually exclusive, of course, and so both could be occurring. But yes, the transport hypothesis is definitely one of our thoughts there.
Thanks, Matt.
Anybody else? I'm curious of what you see as the next steps and how you see yourself proceeding from here.
Well, I laid out some of the next step with the Passerine study, and I think that the effects characterization that will happen over the next 2 to 3 years in that study will be very important. And so that's really what we're focused on in my lab here at EPA, is in making sure that we're ready to do that characterization of effects, basically. You know, there's a world of work out there to do. I spent, you know, an hour just talking to you about one of 9,000 chemicals. There are an awful lot of other PFAS out there that are worthy of consideration, especially as some of these older chemistries, PFAS, PFOA, are phased out of production in some newer chemistries. We're making different perfluorinated chemicals now. And I think a focus on some of those newer chemistries would be very interesting as well.
Not to mention the fact that PFAS, which has been the greatest chemical out there, is when it does break down, which isn't very much, but when it does break down, it breaks down to other PFAS chemicals.
Right. Right. The degradation of these chemicals in the environment is very complicated. And as Bill aptly pointed out, they tend to produce other PFAS as they do so. And that can complicate our not just our understanding of toxicity, but our understanding of exposure as well. I think another very interesting question in this research is, and this is always the case with environmental toxicology, is what are the effects of exposure to mixtures of these chemicals, right? We're sort of in the mode of looking, well, does PFAS matter? Yes, no. Does this other thing matter? Yes, no. But the birds aren't exposed to just one of them. They're exposed, as you see, to a whole host of these things. And how do these things behave together? Is it an additive effect? Is it a synergistic effect, an antagonistic effect? You know, those are extremely complicated questions to try to ask and understand in environmental toxicology.
You're getting compliments on a good presentation. One person says, I learned a lot, but didn't know enough to ask any questions.
Well, thank you for coming.
I know how that feels. The whole thing makes me wonder how far along we are in trying to eliminate these toxins. It doesn't sound like we're working on it very hard.
I think we are. I mean, you know, if you look, so if you follow that SERDP diagram that I presented in the penultimate slide there are whole groups of investigators exploring non-PFAS AFFF, for example. aqueous film forming foams or non-PFAS firefighting products. And so people really are looking for substitutes for some of these chemicals in their various applications. But it's very challenging. I mean, the strength of that carbon fluorine bond and some of the other properties of these chemicals makes them very useful for a lot of the industrial processes that we rely on them for. And finding substitutes is not necessarily easy.
Well, it's at least good to hear they're working on that piece of the puzzle because, yeah.
Yeah, there's great studies in there. There's also studies on how to take the stuff out of the environment once it's in the environment, and so on. So anyone with an interest, I would really encourage you to go check some of those out.
All righty. Anybody else got questions or comments or thoughts?
Thank you very much, Matt. That was very good.
Thanks, Bill. Thanks for coming. I was glad to see you here. And thanks all for coming. I appreciate your attendance.
Much appreciated.
Said something about how as a bird was excreting urine, it could be reabsorbed. How does that work?
Well, it just happens through the tissue membranes, I believe, and they're just reabsorbed back into the body of the bird and go back into circulation.
So it's not really going out.
That's correct. For some PFAS that happens, that the kidneys initially remove it, and as it's being shunted out, it's then reabsorbed through the membranes of those tissues as it's moving.
Well, thanks everybody for coming out tonight. Matt, do make sure you make me the host again so that the recording doesn't get all messed up here.
Sure, I'll do that right now.
Thanks,
There you
Matt.
go.
Great
Yeah,
talk.
thanks
Thanks.
so much.